Precious coral, treasured throughout the world since ancient times. Japan is renowned as one of the world's leading production centers. And yet the ecology of precious corals and their mode of life have been shrouded in mystery. In recent years, developments in scientific technology have gradually enabled us to shed some light on that mystery. This is probably the kind of scene or image that springs to mind when you hear the word coral. But these are reef-building corals, which live in shallow water bathed in sunlight. While reef-building corals and precious corals both belong to a group of aquatic animals called cnidarians, precious corals are solitary, living in deep water. Precious coral fishing began in Japanese waters in the 1870s, when it was already known that these creatures live only in the deep sea. But it wasn't until the 1970s, with the development of deep-sea research equipment, that it became possible to observe them living in their natural habitat. Full-scale scientific research on precious corals began even later, from the year 2000. Precious corals do look like plants, but they are actually animals. They have a hard internal skeleton called the axis. This is the part that is valued as a gemstone. The axis is covered with a fleshy layer of tissue called the senenchyme. In the senenchyme, tiny organisms called polyps carry out the coral's vital functions. The tentacles here have just trapped some prey. These tentacled polyps, called autozoids, eat and digest the coral's food. Each polyp is an individual animal. So the coral is actually a colony made up of numerous polyps. Precious corals also have another type of polyp, too small to be seen with the naked eye. This is an illustration of the senenchyme. The polyp here is different from an autozoid. It is smaller and has no tentacles. It is called a siphonozoid. Siphonozoids supply water to the whole colony through tubes called selenia. Autozoids always have 8 tentacles. So within the classification of cnidaria, precious corals are members of a group called octocorals. In contrast, the polyps of most reef-building corals have multiples of 6 tentacles. These belong to a group called hexacorals. The hard skeleton of both types of coral is mainly calcium carbonate. However, their crystal structure is different. Precious corals are calcite. Reef-building corals are aragonite. So precious corals and reef-building corals differ in their body structure and ecology. But how different are they from a genetic point of view? Koji Uda of Kochi University has examined the genome sequences in the cell mitochondria of both types of coral. Genetic information in mitochondria is different from that of the nucleus. Mitochondrial DNA is composed of only around 18,000 base pairs. This is much fewer than nuclear DNA, so it is easier to map all the base sequences.
Reef-building corals belong to a subclass of creatures called hexacoralia. Precious corals belong to a subclass called octocoralia. The difference is only a subclass, but if we think of this in human terms, it amounts to the difference between a human and a platypus.
This diagram represents the genetic relationship between mammals. The first mammals on Earth were closely related to members of the platypus family. That was about 200 million years ago. These then diverged into many different branches before the ancestors to humans appeared between 6 and 7 million years ago. So although humans and platypuses are both mammals, they are far removed from each other.
So, as for the difference between the mitochondrial genomes of reef-building corals and precious corals, about 70 to 75% of their base sequences are the same. In other words, about a quarter of them are different. Humans and platypuses also share about 70% of their base sequences. So we know from looking at their mitochondria that the difference between hexacorals and octocorals is about the same as the difference between humans and platypuses.
This is a mitochondrial genome map of precious corals as determined by Dr. Uda. Because mitochondrial genomes evolve more quickly than nuclear genomes, they are more useful for the analysis of relationships between related species. There are 8 known species of precious coral in the world today. In the waters around Japan and between Japan and Taiwan, there is Japanese red coral as well as pink coral and white coral. In the Mediterranean, there is red coral. In Hawaii and around Midway Atoll, there are 3 other known species, one of which has yet to be given a scientific name.
Due to their morphological characteristics, Japanese red coral was originally identified as a member of the genus Paracorallium, while Mediterranean red coral was identified, along with white coral and pink coral, as the genus Corallium. I analysed the mitochondrial genome sequences of these 4 species of precious coral, and I found that the DNA of the 2 red coral species varies by only 100 bases. But when I compared Japanese red coral with white coral, and then again with pink coral, I found a variation of about 700 bases in both cases. This detailed comparison of the 4 species clearly showed that Japanese red coral and Mediterranean red coral belong to one group, while white coral and pink coral belong to another. Recently, it's been proposed to classify Japanese and Mediterranean red corals as the genus Corallium, and the white and pink corals as the genus Pleurocorallium.
A gene-based classification system for precious corals has now been established. New information has also been uncovered about their ecology. Seong-won Lee of Risho University is studying the stable isotope ratio of elements such as nitrogen and carbon in body tissue. In the process of metabolism, the light stable isotopes in the food animals eat are excreted from their body more quickly. The heavier the stable isotopes are, the longer they remain in the body. The proportion of heavy stable isotopes is therefore higher in predatory animals than in the food they eat. What do precious corals eat, and by what are they eaten?
Here are the results of an analysis of some Japanese red coral samples collected from the sea around the Ogasawara Islands. Between phytoplankton, or particulate organic matter, and the coral, the carbon stable isotope ratio has risen 1 part per thousand, and the nitrogen stable isotope ratio has risen 2.5 parts per thousand. Furthermore, in this species of sea snail that attaches itself to Japanese red corals and feeds off them, the carbon stable isotope ratio has risen 1 part per thousand. and the nitrogen stable isotope ratio has risen 2.1 parts per thousand compared with the coral. These results suggest that Japanese red corals eat phytoplankton or particulate organic matter, and the corals are eaten by a species of sea snail, indicating an ecosystem in which the Japanese red coral is a part of a food chain.
What is the secret of their colour? It was phytoplankton, the food of precious corals, that held the key. Hiroshi Hasegawa of Kanazawa University is pursuing that mystery.
Corals can't produce red pigment by themselves. Corals take in organic matter that falls to the ocean floor. When the organic matter contains plant components, precious corals are thought to assimilate the geranyl-geranyl diphosphate that is found in plants and use that as the raw material to synthesize carotenoid pigments inside their body.
For this reason, the semenchyme of every precious coral species looks red. However, the colour of the axis varies according to the species.
When precious corals are alive, all the species, white, pink and red, are more or less red. But for some reason, when the axis is taken out, white coral is white, pink coral is pink, and red coral is red. So, regarding the colour of precious corals, the biggest mystery left to solve is how the red pigment, canthaxanthin, becomes fixed inside the axis. That's something we still don't know.
Spring 8, one of the world's largest synchrotron radiation facilities, has enabled researchers to investigate how the axis of precious corals is formed. By generating synchrotron radiation 10 billion times brighter than the Sun and focusing it on a material, they are able to explore the material's structure and composition. Yoshimasa Urushihara of the University of Hyogo used Spring 8 synchrotron radiation to examine the semenchyme of white coral with a CT scan. Bony fragments, called sclerites, are found in the semenchyme. Dr. Urushihara examined the distribution of sclerites in this dried piece of white coral. The autozooids are retracted within the semenchyme. This is a three-dimensional reconstruction of an image from the CT scan. Tiny grains are packed closely together on the surface. Each one of these is a sclerite. Dr. Urushihara's research enabled more detailed observation of the way in which the body tissue of precious corals is supported by these densely packed sclerites. When viewing the composition of the interior, sclerites can be seen around the axis. Look at the coloured areas at the tip of the axis. Close observation has revealed that the axis is initially formed from a concentration of these sclerites. Later, the axis epithelium, which forms the outer covering of the axis, begins to produce calcite, and the axis continues to grow. At Spring 8, a research team led by Yasuki Tamamori of the Japan Synchrotron Radiation Research Institute conducted a fluorescent X-ray analysis of a pink coral axis. This revealed the secret of the pattern resembling growth rings that appears in a thin cross-section of a coral axis. The researchers examined the distribution of organic sulfur, inorganic sulfur, magnesium, and phosphorus in the axis at different stages of its growth. The left-hand side of this image is the surface of the axis. The further to the right, the deeper into the interior. The redder the colour is, the higher the concentration. That is, the larger the quantity that has been assimilated. At the other end of the scale, blue indicates a small quantity. If you look inside the white frame, you can see that magnesium is deep red, while inorganic sulfur is yellow and phosphorus is light blue. So at times, when a large quantity of magnesium is assimilated, the quantities of inorganic sulfur and phosphorus are small. Red areas of high concentration are seen here to alternate with yellow or blue areas of low concentration. In other words, we now know that the axis assimilates trace elements periodically as it grows. And since the darker coloured growth rings coincide with high concentrations of inorganic sulfur, we know it must be the inorganic sulfur that forms the rings. How fast do precious corals grow? Masatoshi Yamada of Hirosaki University observes the growth rates of precious corals by measuring radioisotopes.
Among natural radionuclides, the half-life of lead-210 is 22.3 years, so it can be used for dating back around 100 years. We're measuring the concentration of lead-210 by measuring the alpha rays of its daughter nuclide, polonium-210.
Professor Yamada analysed the base of this pink coral. Here are the results. The concentration of lead-210 is high on the surface of the axis because it has just been assimilated from the seawater. The concentration of lead-210 decreases exponentially with time after it is assimilated. The rate of this decrease was used to estimate the growth rate of precious corals.
So, how long does it take a precious coral axis to grow to the size of a human adult's little finger? The answer is about 50 years.
In other words, we now know that Japanese precious corals grow only about 0.3 millimeters a year in diameter. Science has now confirmed just how rare it is for a large colony to exist. Slow-growing Japanese precious corals. It takes a long time for a large colony to form. A comparison with the Mediterranean, another natural breeding ground, revealed another characteristic of Japanese precious corals. In 2009, Giorgio Bavestrello of the University of Genoa and Riccardo Cattanovietti of the Polytechnic University of Marche joined the study from Italy. After looking at live corals and their distribution on the ocean floor, they pointed out a difference between the corals in Japanese and Mediterranean waters.
The difference is the density of the population. In the Mediterranean, in shallow water population, we reach densities about 600-800 individuals, colonies for square meter. This seems not reached in this water by this species. The density of the species are very low, respect those we have we have observed in the Mediterranean.
The population density of Japanese red corals is only 0.005 colonies per square metre, while that of the Mediterranean red corals is over 40 colonies. It seems that population density varies among species. If we hope to continue exploiting precious corals, we must develop effective methods of resource management. To manage living aquatic resources, it's important to understand their ecology and, above all, to know how they breed. Kazuo Okuda of Kochi University has calculated the reproduction period of red corals living off the coast of Kochi Prefecture. The waters off Kochi are one of Japan's largest natural breeding grounds for red corals. After re-examining previous experimental methods, Professor Okuda developed a method of dissolving only the sclerites in the coenenchyme. This has made it possible to strip the entire coenenchyme off the axis in one piece, just like removing a piece of clothing.
The coenenchyme that was attached to the axis is living tissue. Here it is after being decalcified and stripped off the axis.
Mature Japanese red corals were identified for the first time in this way among samples from the waters off Kochi.
What we have here is an egg. There are some tentacled polyps over here, but the egg's not inside them. It's in a different place. In fact, this is one of the tiny polyps without tentacles. It would normally be smaller, but now it's all puffed up and swollen with the egg inside it.
According to Professor Okuda's research team, we now know that the red corals living off the coast of Kochi are dioecious. The sperm-producing male corals and the egg-producing females were found to be separate colonies. Research has also found that reproductive germ cells were produced inside the siphonozoids, the polyps without tentacles. Previous research has found that Mediterranean red corals produce germ cells in their autozoids, so this is a significant difference between the two species. Japanese red corals produce not only germ cells, but also sperm in their siphonozoids. Another characteristic that has come to light is the reproduction period of Japanese red corals.
When red corals living off the coast of Ashizuri, in Kochi Prefecture, produce their reproductive germ cells, it's so well timed, I believe it's seasonal. We've been monitoring them year-round for 5 years, and we've found that they generally produce germ cells in March, April and May. It's concentrated around that time. We measure the temperature of the water at a depth of around 100 meters at the same time, you see. And the timing is right around March, just after the temperatures have reached their lowest and the seabed is about to start warming up again. The seabed is coldest in January and February, and March is just when the temperatures are about to start rising. That's when the corals start producing germ cells.
The findings of Professor Okuda's research team were immediately reported to the government of Kochi Prefecture, who responded by prohibiting coral fishing in Kochi during the months of June and July. This is the first time a closed season for coral fishing has been tried in Japan. Further research is needed to ensure future supplies of Japanese precious corals. Nozomu Iwasaki of Risho University has been studying the distribution of precious corals in Japanese waters for over 10 years.
I studied 2 sea areas inhabited by Japanese red corals. One area where the corals had already been harvested and one that hasn't been fished yet. When I compared the size and age of the red corals in the 2 areas, I found colonies of various sizes, from large to small in the unfished area. But only small colonies were left in the harvested area. We learned from comparing these 2 areas that it takes 10 to 20 years for a small colony to grow to a reasonable size. We can use this information to work towards a sustainable fishery. In other words, if we close an area to fishing for 10 or 20 years after each harvest, the corals will have time to recover.
The range and scope of the precious coral study is widening with the participation of students who will lead the next generation of research.
Precious coral is admired for its beauty and prized as a gemstone. In addition to its beauty, I would now like to suggest 2 new criteria for managing this valuable resource. that precious corals are harvested in harmony with the natural environment, and that they are harvested by sustainable fishing methods. It's up to us to conserve our resources of precious corals and hand them down to future generations.